Good morning, students. So we will continue with the same topic, Chara, and today we are going to discuss about the fertilization process in Chara. पिछली क्लासेस के अंदर we have discussed about the reproductive specialized या peculiar reproductive structures which are found in Chara, that is globule and nucleole. In today's class, we'll discuss how the fertilization takes place in Chara. So as we all know that during the fertilization, the tube cells just below the corona get separated slightly and form five narrow slits or openings. हमने पिछली क्लास के अंदर जब पिछली क्लासेस के अंदर हमने न्यूक्लियल की स्ट्रक्चर का स्टडी किया था तो हमने देखा था कि न्यूक्लियल के अंदर डिस्टल एंड की तरफ हमें कोरोना सेल्स हमें देखने को मिलती है. They are 5 in number, and these 5 corona cells they get separated when it is matured, and वो नैरो स्लिट्स का निर्माण करती है. और एंथ्रोजोइड्स जो कि ग्लोब्यूल के अंदर जिनकी फॉर्मेशन होती है, they are bioflagellated and they pass through these slits and they enter into the nucleole. Out of the many aggregated anthrozoids toward the slits, only one comes near the receptive spot of the egg. यानी सिर्फ एक ही जो receptive spot होता है egg का, जो कि egg which is present inside the nucleole और tube cells के जो जिसको tube cells protective covering के रूप में घेरे हुए रहती है, अंदर उसके egg होता है. तो receptive spot की तरफ एक ही हम कह सकते हैं anthrozoid उसके नज़दीक पहुंचता है. On contact with the egg, it fuses and forms an oospore. यानी haploid जो structures which are called as a gametes, उनकी synagamy हमें fertilization के अंदर देखने को मिलती है. या this step is called as a synagamy, the process of the fertilization, and both the male and the female gamete fuses and they form a diploid structure that is called as a sporophyte, या that is called as a oospore particularly. जब हम oospore की बात करते हैं तो इट इज़ हार्ड स्पेरिकल टू एलिप्सोइडल इन शेप एंड वेरियस कलर्स लाइक लाइट येलो, ब्राउन, रेड और ब्लैक. इस तरह का वेरिएशन उसके अंदर हमें देखने को मिल सकता है. इट इज़ सराउंडेड बाय 4 लेयर्ड वॉल्स ऑफ विच द आउटर 2 आर कलर्ड एंड इनर 2 आर कलरलेस. यानी 4 लेयर्स हमें देखने को मिल सकती हैं उसके अंदर, जिससे इनमें से द आउटर 2 आर कलर्ड, जो बाहर की तरफ जो है 2 कलर देखने को मिल सकती है, इनर 2 आर दे आर कलरलेस. नाउ germination. During germination, the nucleus of the oospore migrates toward the upper region. After fertilization की प्रोसेस कंप्लीट होने के बाद में उसका जर्मिनेशन हमें देखने को मिलता है. और जो oospore का जो न्यूक्लियस होता है, it migrates toward the upper region. The nucleus then undergoes meiotic division to form haploid nuclei. That is oospore का nuclei that represents the sporophytic phase, अवस्था को रिप्रेजेंट करता है. और अब इसके बाद में it undergoes meiotic division to form 4 haploid nuclei. The oospore then divides into 2 unequal cells, of which the upper lenticular cell contains 1 nucleus and the lower large basal cell contains 3 nuclei. The nuclei of the basal cell gradually disintegrates, degenerates या disintegrate, खत्म हो जाता है. Now this is a slide representing the successive stages in oospore germination. You can clearly see that is diploid nucleus is there. Diploid nucleus that is which is covered by oospore wall, and this diploid nucleus undergoes meiotic division. It results in formation of the haploid nuclei. They are 4 in number. और बाकी जो it is one is cut off from the 3 others and it is placed in a lenticular cell, and the 3 disintegrate या degenerate हो जाते हैं, degenerating nuclei, and the lenticular cell के अंदर जो nuclei पाया जाता है, it starts germinating. It makes protonemal cell की तरफ behave करता है. डिवीजन होता है, राइजोडल सेल बनती है उसके अंदर, एंड द एक्सिस इट स्टार्ट्स कमिंग आउट, ये डिवीजन होता है, इट रिजल्ट्स इन फॉर्मेशन ऑफ द एक्सिस, और उस एक्सिस के ऊपर हमें नोड्स एंड इंटरनोड्स एंड राइजोइड्स और इस तरह के स्ट्रक्चर्स का डेवलपमेंट हमें उसके अंदर पर देखने को मिलता है. सो दिस इज आई हैव टेकन ए डायग्राम, ए stained slide या stained picture of oospore. Yeah, it's better to say that the structure of the globule, sorry, it's a structure of nucleus जिसके अंदर it is after fertilization. ये इसके अंदर ये different, ये matured condition के अंदर when the fertilization has taken place. Now the lenticular cell projects out by rupturing the oospore wall and divides mitotically by an oblique longitudinal septum to form a larger protonemal initial and a small rhizoidal initial. जैसा कि स्लाइड के साथ मैंने डिस्कस किया था कि लेंटिकुलर सेल, बाकी तीनों सेल, तीनों न्यूक्लियाई डिसइंटीग्रेट या डिजेनरेट हो जाते हैं. उसके बाद में जो लेंटिकुलर सेल है ये माइटोटिकली डिवाइड करती है और it forms protonemal initial and the small rhizoidal initial. Both the initials grow in the opposite direction. Rhizoidal initial results in the formation of the rhizoids. The protonemal initial is differentiated into nodes and internodes and form the upper part of the plant body, whereas the rhizoidal initial forms the rhizoids. And then the secondary rhizoids may develop from the lower node of the protonemal filament. So this is how the germination of oospore in Chara takes place. And now next slide is about the life cycle of the Chara. इसके अंदर it is represented diagrammatically कि what are the various modes of the asexual reproduction जो Chara के अंदर हमें देखने को मिलता है, by which it completes its life cycle, that is by secondary protonema, amorphous bulbils, amylum stars that we have already discussed during the phase of the asexual reproduction in Chara. And when it comes to the sexual reproduction that we have discussed in a very descriptive manner, that by the formation of the globule and nucleus the sexual reproduction takes place, that is by formation of the male gametophyte, female gametophyte, male gametangia, female gametangia, the specialized structure, male reproductive structure, female reproductive structure, which are called as a globule and nucleus, which are being formed, and in them there is formation of the anthrozoids and egg cells and their fertilization that results in the formation of the zygote, and the zygote undergoes meiotic division. It results in the formation of the protonema, and the protonema makes the plant body again. So that is how the life cycle of Chara is completed. So this is another diagram. I have taken one more slide to explain the same. That was, it is diagrammatically. In this you can see Chara plant is there, globule is there, nucleus is there. It depicts the same basic concept of the life cycle of Chara that I have already discussed. This is one more diagram, this is one more slide. Thank you very much.